For small organisms, such as amoeba, diffusion alone is enough to meet the requirements of the body, that is, to supply nutrients and oxygen, and also the removal of waste products. This is possible because substances travel small distances so that they are moved in and out of these small organisms. But for larger organisms that are more active, they cannot rely on only diffusion because their cells are often deep within their bodies, which means that they have to travel some long distance so that the materials are moved in and out of these cells. Now, due to the fact that the cells are more metabolically active, the supply of nutrients and oxygen must be rapid, and at the same time, the removal of waste products must also be rapid. Now, what has happened is that these organisms have developed well transport systems with pumps to keep the movement of fluids around and through them so that the materials are moved in and out of the cells. So let's look at our main interest, which is the mammalian cardiovascular system. The mammalian cardiovascular system is made up of a pump, which is the heart, and blood vessels. Within the blood vessels, we have the fluid, which is known as blood, and it travels within these vessels. The travel of blood within the vessels is known as a saturation. And within the cardiovascular system, we have what we call two types of saturation. That is the systemic saturation and the pulmonary saturation. The systemic saturation is whereby blood is pumped out of the left ventricle into the aorta and then travels to all parts of the body except the lungs, and it returns to the right side of the heart within the vena cava. Simply meaning that the systemic saturation is where blood moves to all parts of the body excluding the lungs. The pulmonary saturation, however, is where blood is pumped out of the right ventricle into the pulmonary arteries, which carry blood to the lungs. Then, from the lungs, blood returns to the left side of the heart through the pulmonary vein. That is the pulmonary saturation. In brief, that is saturation of blood to the lungs and its return to the heart through the pulmonary vein. The combination of the pulmonary saturation and the systemic saturation makes a double saturatory system, which implies that the cardiosaturated system or the cardiovascular system is a double saturatory system. We earlier said that the mammalian cardiovascular system is made up of the heart and blood vessels. Let's look at the blood vessels. There are three main types of blood vessels. We have arteries, veins, and capillaries. Arteries, vessels carrying blood away from the heart. Veins are vessels carrying blood towards the heart. Capillaries are vessels linking arteries and veins. These capillaries take blood to almost every cell within the body. Let's have a look at arteries. The function of arteries is transport blood at high pressure to tissues. The structure of the wall of an artery enables it to perform this function efficiently. Arteries have walls that are made up of three layers. We have the tunica intima, in brackets the inner endothelium, which is made up of a layer of flat cells fitting together, and this layer is very smooth, minimizing friction within the moving blood. The other layer is the tunica media, in brackets the middle layer, which contains smooth muscles, collagen and elastic fibers. The third layer is the tunica externa, in brackets outer layer, containing elastic fibers and collagen fibers. One of the distinctive characteristics of an artery wall is its strength and elasticity. Due to the fact that blood leaving the heart travels at high pressure, the artery walls must be extremely strong to withstand this pressure, and this is achieved by the thickness and composition of the artery wall, so that they can provide great strength and resilience to the high pressure. In other words, the one way of distinguishing or differentiating arteries from the rest of the blood vessels is by looking at the thickness of the artery wall, whereby the artery walls are more thick compared to the rest of the blood vessels. The tunica media, which is the middle layer, is the thickest part of the wall. It contains large amounts of elastic fibers which allow the wall to stretch as the pulses of blood surge or increase at high pressure within the lumen. The elasticity of the artery walls reduces the chances of bursting when the pressure becomes high during its movement. This elasticity also has another function. When the ventricles contract, they push blood out of the heart at high pressure, and this pressure reduces when the ventricles relax. Now, when the artery wall stretches at high pressure, it is going to recoil as the pressure lowers. During recoiling of the artery wall, they give a small push to blood within the lumen, increasing its pressure a little, even when the ventricles have relaxed, meaning that they try as much as they can to maintain the high pressure. Maintaining this pressure is brought about by the recoiling of the artery walls, which give a small push during recoiling, and this small push is inserted on the blood, increase the pressure. As arteries reach the tissues to which they are transporting blood, they branch into smaller vessels called arterioles. These arterioles have their walls with a greater proportion of smooth muscles. They also have small traces of collagen fibers and elastic fibers. Having smooth muscles gives them the capacity to contract, narrowing down the diameter as they supply blood to the tissues. They can also dilate to increase blood supply to the tissues depending on the oxygen demands. Take for instance, during exercises, when there is a greater oxygen demand, there is increased supply of blood to the tissues. What happens to the arterioles is that they are going to dilate to increase the lumen, and in that way the blood flow to these tissues increases. But at rest, when the oxygen demand for the muscles is very low, they contract, reducing blood flow to the tissues. As blood flows along the arteries, blood pressure reduces significantly. If we introduce figures, as blood is pumped out through the aorta, its pressure is around 120 millimeters of mercury, that is around 16 kilopascals. And as blood reaches the arterioles, its pressure is around 85 millimeters of mercury, which is 11.3 kilopascals. And this will significantly continue reducing as it flows through the capillaries. And by the time blood gets out of the capillaries, it is around 35 millimeters of mercury, which is around 4.6 kilopascals. All in all, what we have to know is that as blood flows along the arteries, there will be that reduction in pressure. And by the time even it reaches the veins, the pressure is going to be very low. Let's have a look at the capillaries. We said that arterioles branch off from the arteries. Now, these arterioles continue to branch, forming the tiniest blood vessels that are known as capillaries. The function of capillaries is to take blood as close as possible to all cells, allowing rapid transfer of substances between cells and blood. So what capillaries do is that they take blood as close as each and every cell within an organism's body. And once they take it there, we shall have exchange of materials between blood and the cells. As capillaries reach every tissue in the body, they form a capillary bed, which is a finest network connecting all the tiny capillaries within those tissues to allow efficient exchange of materials between the cells and blood. The capillaries are small in size, and their diameter is almost as the same as that of red blood cells, which makes them able to bring blood as close as possible to each group of cells. The walls of capillaries are extremely tiny, and these walls are made up of a single layer of endothelial cells. Recall that we have said that the diameter of each capillary is similar to the size of red blood cells. Remember that the red blood cells have a component of hemoglobin in them that transports oxygen. That's why they have to reduce almost to the size of a red blood cell, so that each red blood cell is as close as possible to a cell so that it can give out the oxygen to the respiring tissues or to the respiring cells. And as these red blood cells squeeze through these capillaries, that's when they are going to be close. That's when they are going to be brought close to each and every cell that needs oxygen, and then the exchange of materials can take place. In most of these capillaries, there are tiny gaps, or what I can call tiny pores, between the individual cells that form the endothelium that lines the capillary wall. These pores allow some blood components to seep or pass through them into the space which is between the blood cells and body cells, forming a tissue fluid. When the materials seep out of the capillaries, they get into that space which is between the capillary wall and then the blood cells. Now, when they get into this space, they form what you call a tissue fluid. Now, we have to know that all body cells are always embedded in this tissue fluid. We shall get back to tissue fluid formation in the following tutorials. Let's have a look at the veins. As capillaries reach the tissues, we say that they branch into more finest vessels to form a capillary bed, and it is these finest vessels that are now going to again join up to form larger vessels known as venules. The venules later join to form a major vein, or they form veins. The function of the veins is to return blood to the heart. All veins in the body return blood to the heart. Remember, we said that by the time blood gets to the capillaries, its pressure has drastically reduced. The reduction in pressure is to allow or give more time for the exchange of materials between blood and tissue cells to take place, which implies that by the time blood enters the veins, the pressure is very low. And due to this low pressure, the veins do not need to have a thick wall. They have a thin wall made up of 3 layers: tunica intima, tunica media, and tunica externa. Of all the three layers, the tunica media is much thinner and has few elastic fibers and collagen fibers as compared to the tunica media of capillaries, which we saw that has more elastic fibers and collagen fibers. The low pressure within the veins creates a problem, especially within the flow, in that the fact that the blood flows at low pressure, there is that tendency of flow back or blood flowing backwards. In some veins, they are adapted to make sure that they control blood backflow by having valves. By having valves. The function of these valves is to maintain a one-direction flow of blood. The valves are formed from the endothelium. Most of the veins have these valves to maintain one direction flow for blood. When you find that some have the semilunar valves that fill up when blood tends to flow backwards. So when they fill up, blood piles up on the opposite direction due to the flow, and then this forces the valves to open, thus maintaining one direction. Some of the veins are embedded deep within the muscles, such that when the tense on these muscles increases, or when these muscles contract, they squeeze the veins inwards, creating a temporary pressure that is going to force blood to move forward. Conclusively, all valves, whether in the heart or in the veins, are meant to maintain a one-direction flow. Look at the semilunar valves at the base of the aorta. As blood is pumped out at high pressure through the aorta, when the ventricles relax, there is tendency of blood flowing out of the aorta back into the ventricles. But the semilunar valves are going to close, and as they close, blood does not flow back to the heart. So when the ventricles contract again, that pressure is going to force these semilunar valves to open again, allowing blood to flow out of the heart. We also have the semilunar valves at the base of the pulmonary artery that prevent blood from flowing back when the ventricles relax. That was to do with our blood vessels. Just having a sample questions here that we can try out, and we give answers in the comment boxes where other teachers and tutors will cross-check and give appropriate answers. The first question is, explain the roles of the sino-arterial node, in brackets SAN, the atrioventricular node, which is AVAN, and the pubic tissues during heartbeat. The second question is that, suggest the reason why The wall of the base of the aorta is more thicker than the wall of an artery, getting close to the tissues. Thank you. Continue to subscribe and share. Let's learn more. All the best.